Hello, good morning, my dear students. Myself, Mr. S.C. Gangavane, Assistant Professor, Department of Biotechnology, Coxid, Latur. Today we are learning about one more interesting topic regarding the classification of viruses in virology. So let's start. Please give concentration to here. So first, the definition of viruses. Okay, definition of virus. What is virus? So viruses are the small obligatory parasites. Viruses are the small obligatory parasites which require the host cell to carry out the life cycle. So then and then virus can live inside the host cell. Okay, that is we call as a virus. It is a very tiny small particle we cannot see without a microscope. Okay, so that's why the virus is an obligatory parasite, means strict and strict. It require what? The host cell. Without host cell, virus cannot be show the replication. Okay, so that's why that is a virology in that the virus definition. Next one, come to the next point, that is a classification of viruses. In the classification of viruses, there are different characteristics takes under the consideration when you classify the virus into different categories or the groups. So one by one we will see. So first category is that the viruses are classified into different groups on the basis of various characteristics. In that, the first one is the on the basis of host. So on the basis of host, the viruses are classified as animal or plant viruses. Second one is the bacterial viruses or bacteriophage, or animal or human viruses. So first one is the human or animal virus, second one is the plant virus, and third one is the bacterial virus. Okay, so human and animal viruses mostly infect to the human host cells, and it will cause a disease after the infection, after the pathogenicity. So such viruses we can see here the example polio virus. We know it causes the polio in humans. So polio virus, herpes virus is also the human virus which will infect the human. So it will infect mostly the brain cells. So cephalitis is caused by the herpes virus, and another is the herpes virus 2 that causes the infection to the genital organs. So it is sexually transmitted diseases due to what? It is due to the herpes virus. So it is responsible for to transmit the STD diseases. Okay, so from normal person is infected by what? It is infected by the infected person when they both are coming to contact or sexual intercourse. Okay, so then is the plant viruses. In the plant viruses, we can say that these viruses are going to infect to the plant cells, and they show their replication or multiplication or assembly in that plant. So the plant virus examples is the TME virus, then the CME virus or CAME virus. So TME virus means tobacco mosaic virus, CME means CAME means cauliflower mosaic virus, and the CME means the cucumber mosaic virus. So in this way, the viruses which belongs to the plant family or which infect the plant. Okay, so then bacterial viruses are also called as the bacteriophages, and that bacteriophages are different types. So bacteriophages, they infect to the bacterial cells, and it shows the different type of the life cycle, lytic cycle and lysogenic cycle. So that bacterial viruses also called as bacteriophage means for example, we can say the lambda phage is the example of a bacterial phage or bacteriophage or bacterial virus. Then these bacterial viruses or bacteriophages are also classified into two types, that is T even bacteriophages and T odd bacteriophages. So T even bacteriophages are T1, T3, okay, T even are T2, T4, T6 and T even are T1, T3, T5. In this, the odd numbers and even numbers, on the basis of that, what happens, on the basis of that, the viruses are classified that T even viruses. So these are the examples of bacterial viruses, definitely they infect. So the human viruses show the icosahedral symmetry or spherical shape in structures. Plant viruses are showing the helical symmetry, or they looks like in rod shape, and the bacterial viruses, they looks like the or they show the complex symmetry. Okay, so in this way we can study the different types of the structures of the viruses under the electron microscope. Okay, so let's go ahead. And second point, so on the basis of symmetry of structures, means viruses are classified on the basis of symmetry of structures, in that the icosahedral symmetry or polyhedral symmetry or the spherical structure of viruses we can found. Okay, so example is the hepatitis virus. Okay, when you see the hepatitis virus under the microscope, definitely that hepatitis virus is the show the infection to the humans and mostly the animals. So that's why such viruses belongs to the polyhedral or spherical or icosahedral symmetry, means they show such type of structures. Then next one is the helical symmetry, showing viruses or rod shaped structures, viruses on the basis of morphology. So another example of this helical symmetry is the TMV, tobacco mosaic virus. This tobacco mosaic virus is showing the helical symmetry or structure. It means that what, this tobacco mosaic virus first time found on the leaves of tobacco, okay, and it forms the black color spots. So from that we can isolate such type of tobacco mosaic viruses and we can find out its structures, and that structures it looks like the helical structures, it looks like the rod shaped. We will discuss later in detail that structure. So still just come to the next point, that is the complex symmetry or tadpole like structures. So complex symmetry or tadpole like structure showing viruses belongs to the bacteriophages, means the bacteria. So bacterial viruses, that is called as bacteriophages, they show the complex symmetry and they looks like tadpole like. So that's why it is called as the tadpole like structure of that complex symmetry showing bacterial viruses. Example is lambdaphage, we can take for consideration. Lambdaphage, when you see under the electron microscope, definitely it will show the complex symmetry like this. Okay, so we will see in the latter. Okay, so next one is the on the basis of the viruses are classified on the basis of its genetic material. Some viruses possess the DNA as the genetic material, some viruses possess the RNA as the genetic material. So on the basis of DNA and RNA, the viruses are further classified. Okay, so the first one is the DNA viruses. The viruses which bears the DNA as a genetic material, that is called as the DNA viruses, and DNA viruses are further classified into two types. One is the double-stranded DNA viruses and another is the single-stranded DNA viruses. The double-stranded DNA virus means what? It is nothing but it possesses the double strand. So that's why it is a double-stranded DNA viruses. If the virus possesses a single strand of DNA, so it is called as the single stranded DNA viruses. Simply like that of the RNA viruses also bears the RNA. So if it is a double stranded RNA, then double stranded RNA viruses. If it is a single stranded RNA, then it is called as the single stranded RNA viruses. That's all. So that is a classification on the basis of what? On the basis of its genetic makeup or material. Okay. So next, come to the another point, that is the fourth point, that is on the basis of the sense RNA strands. Okay, viruses are classified on the basis of presence or absence of sense RNA strands. So these are as follows. First one is the positive sense RNA viruses, and that is a positive RNA, or negative sense RNA viruses, that is negative RNA, as a genetic makeup present in their body or in their structures. So in this way, positive sense RNA and negative sense RNA, these are the classification of viruses on the basis of sense RNA. Okay. So next one is the on the basis of envelope. Okay, on the basis of envelope. Envelope is the outermost layer which is present over surrounding, which overs or which surrounds the capsid, and that is nothing but the envelope, and the envelope is nothing but made up of from the different compositions of, and that different composition means it is made up of the mostly polysaccharides. Okay, so enveloped viruses and non-enveloped viruses. The virus which bears, which acquires the envelope, that is called as envelope viruses. The viruses which do not contain the envelope, that is called as the non-enveloped viruses. Simple is that. But if the structure contains the envelope, so enveloped viruses. If doesn't, then it is called as the non-enveloped viruses. So on the basis of nature of receptors, we know that on the envelope or on the capsid there are the receptors which has a different compositions. So every virus possesses the different glycoproteins or lipoproteins on their surfaces to attach the host cell receptors to enter into the host cell. So on the basis of that, on the basis of receptors composition, chemical composition, okay, we can classify the viruses in two groups. That is, one is the glycoprotein receptor bearing viruses and another is the lipoprotein receptor bearing viruses. Okay, means such type of viruses in that glycoprotein is the chemical composition or biomolecules present in the receptor. So that is why that is called as the glycoprotein bearing viruses. If the lipoprotein is the composition of the receptor in the viruses over the capsid or over the envelope, then that is called as the lipoprotein receptor bearing viruses. Simple as that. So next, move on this next point, that is on the basis of virulence or pathogenicity. So viruses are also classified on the basis of their virulence. How much they show the pathogenic effect on the host cell. Okay, so on the basis of that, the viruses are classified. Okay, so that viruses are also called as the virulent phage or temperate phage. Okay, mostly phages are nothing but which show the infection to the bacterial cells. So that bacteriophages are shows the two types of the nature, virulent phage and temperate phage. Virulent phage mostly it is infectious. Okay, it is infectious to host cell. Okay, and death may occur. But the temperate phage which do not show the infection, only genetic material integrated into the host DNA. Means the virulent phage shows the lytic cycle, and during the lytic cycle there is a destruction of the host cell. But in lysogenic cycle, the phage which carry out the lysogenic cycle means only its own DNA adds into the host DNA, then that is called as the prophage, and such type of cycle is called as the lysogenic cycle, because in this cycle there is no destruction of host cell. Okay, so that viral DNA as a prophage remains long time forever when the proper condition gets, environmental factors get, nutrition gets to that particular cell, and then the particular C second protein get degraded, and due to that degradation the lysogenic cycle is converted into the lytic cycle. So such type of temporal phage becomes the virulent phage when the condition is normal. Okay, so next is the on the basis of life cycle. So some viruses show the lytic cycle, means destruction of host cell, okay, mostly, and the another is the lysogenic cycle. In lysogenic cycle only the host DNA is viral DNA is integrated into the host DNA, so that's why it is called as prophage, and the cycle of replication is called as lysogenic cycle. There is no destruction of host cell. Okay, got it? So next one is the on the basis of nucleocapsid. Okay, some viruses bears the nucleocapsid in their structure, ultrastructure, when you see under the electron microscope. Okay, so the viruses which bears the nucleocapsid means the DNA which is attached to the capsid, that is called as nucleocapsid, and such type of structure bearing viruses is called as the nucleocapsid bearing viruses or nucleocapsid viruses. But such structure if not present, okay, means the capsid is absent, only envelope and that contains the DNA, or DNA is not directly attached to the capsid, that time that is called as the what? That is called as the non-nucleocapsid viruses. Okay, non-nucleocapsid viruses. Then on the basis of presence of RT enzymes. What is RT enzyme? RT enzymes are nothing but the reverse transcriptase enzyme. It is a key enzyme. Due to that enzyme, what happens? Due to that enzyme, the RNA is converted into the DNA. Means the viral RNA when enters into the host cell, the RNA is converted into first of all DNA and then double-stranded DNA, and that DNA is integrated into the host DNA. So due to what? Due to the reverse transcriptase enzyme. So that's why the reverse transcriptase is the key important enzyme to classify the viruses. So viruses which bears the reverse transcriptase enzyme, that is called as the RT or retroviruses. Retroviruses, example is HIV. We all know the HIV is a human immunodeficiency virus, and it causes the disease to humans, that is the AIDS, acquired immunodeficiency syndrome. Okay, so in that, and another is the non-retroviruses. Non-retroviruses, example is adenoviruses. It is a non-retrovirus. The non-retrovirus means it does not possess the RT genes, so it will not produce the reverse transcriptase enzymes, and it means that it will not show the replication like the retroviruses. So that's why it belongs to the class which non-retroviruses. Example is the adenoviruses. So these are the classification on the basis of different characters which bears the virus, which characteristics bears by the viruses. Okay, so after that we will see the detailed structure. Okay, in this structure, see here, that is the icosahedral symmetry showing viruses. Examples are mostly animal and plant viruses shows the such type of structures. Okay, so in that the enveloped outside structure which is called as the envelope, inside there is a capsid which is a proteinous structure, and on the envelope there is a receptors. Okay, spikes, that is called as receptors may be the glycoprotein or lipoproteins, and inside the capsid there is a genetic material. It may be the DNA or it may be the RNA. So such type of structure shows the icosahedral symmetry or polyhedral symmetry, or it is also a spherical shape like structures. Okay, so such type of viruses, कोणते viruses दाखवतात? Only the viruses which infect the human or animals. Okay, there may be chances of some animal viruses may infect to the humans also. Okay, so another virus structure is the helical symmetry. In that helical symmetry of TMV, that is tobacco mosaic virus. See, in tobacco mosaic virus there is a tube like structure which is shown by the red color, which is nothing but the halo axis on the hollow axis there is a spherical coiling of the genetic material, and that's why the capsid or capsomere which is arranged which is also in a helical manner. So that's why the structure is called as helical structure of tobacco mosaic virus. So it is the ultra structure of TEMV. When you see under the electron microscope, definitely we will got the this such type of structure. So that is the genetic material, that is the capsomere, it will form the capsid, that is the axis. So such type of things may contain in that structures. So another structure is what the main or important structure, that is it is the combination of icosahedral symmetry and the helical symmetry, because it is more complex than both. Okay, so complex symmetry mostly shown by the bacterial phages or bacterial viruses, that is called as bacteriophage, in that the lambda bacteriophage, and we can take the lambda bacteriophage example, and when you see under the electron microscope, in that see the structure, it is at the top there is a head, and in head there is a DNA or RNA is packed. So DNA is packed as a genetic material. Below that there is a small tapering end that is called as the neck, and that head is made up of from the capsid, and capsid is made up of from the capsomere proteins. So it is a proteinous layer in which the DNA is packed. Below the neck there is a tube-like structure that is called as body or the sheath, and at the base there is a base plate, and on the base plate there is a spikes. Okay, fine spikes that is called, and from the base plate there is a 3 to 4 from two sides. There is a arrangement of the tail fibers for the to hold the host cells, and to enter to break the host cell there is a spikes. So in this way the structure of complex symmetry showing lambda bacteriophage we can find out under the microscope. So that is all about what? That is all about the classification of viruses on the basis of their different parameters or characteristics, and their detailed description about its structures. So that's all, and thank you for joining us.